Let's go.
Hi everyone, my name is Maxime Mahé. I'm a researcher at Inserm, and today we're going to present to you how we can use stem cell and intestinal organoid to study intestinal physiology and diseases. So how can we use organoids to study intestinal physiology and disease? Well, we have been fortunate to have two sources of cells. We can use either iPS cell, induced pluripotent stem cell, or adult stem cells. In the first case, we can reprogram somatic cells from patients and generate human pluripotent stem cells. Those cells can be differentiated. We use a stepwise differentiation protocol where that cells is being differentiated into an endoderm, then a primary gut tube. This primary gut tube will ultimately be cultured in a matrix, and these spheroids that will be formed will produce an intestinal organoid. These intestinal organoids, after 28 days in culture, will form a three-dimensional structure and will bear different compartments. Inside you will see an epithelium with all the intestinal lineages within a lumen that will contain, of course, the luminal sites of the organoids. Interestingly, we also have a stromal compartment that contains mesenchymal cells, fibroblasts, and ultimately muscle cells. This is a very useful model system to understand developmental mechanism of gut growth. In the lab, we use those model system to understand how the human gut is growing, and especially how the enteric nerves or the neurons of the gut are actually playing a role in the development of this gut. We also use that model system to understand developmental disease, or specifically neurodevelopmental disease. And in this case, we can use them to model some congenital diseases, such as Hirschsprung disease. So this is the first model that we can use, derived from iPS cells. But we can also use another type of stem cells. In this case, we will use the adult stem cells that are contained into the gut. Here, this is our gut. We have the lumen, the microbiota, and the nutrients, and we have just beneath the epithelial cells. Within that epithelial tissue, we have in the crypt stem cells. Those stem cells can be harvested. We isolate from patient biopsies stem cells, and we can put them in culture. We put them in an hydrogel, and we let them grow into a three-dimensional structure. The structure that is being formed is also called an enteroid and contain only epithelial cells. These organoids will bear all the epithelial lineage, including absorptive cells or secretory lineages. And this is a really useful model to understand several mechanisms, especially in intestinal disease. And in the context of inflammatory bowel disease, or IBD, we can actually use organoids from healthy patient or patient with IBD and study how inflammation is actually impacting the function of that organoids. In this context, we can study the epithelial barrier function, and we can look how molecules or drugs can go across that epithelial barrier. This model is really also important to generate biobank. Again, we generate those organoids from patient's tissue, and in this context, we can generate biobanks of living tissue. We can either generate healthy control tissue or disease patient tissue and create a bank of organoids that could be used to test drugs and then be used in a context of personalized medicine. So you know everything about intestinal organoids and how we can use them. If you want to know more, please visit our website or follow us on Twitter. Thank you for your time.